Hello, my name is Joshua Mugabi. I welcome you to Biology King. And in this session, we are going to talk about the structure of DNA and RNA. Draw the basic structure of a nucleotide, labeling its parts. Our nucleotide has got a phosphate, it has a sugar, which is a ribose or deoxyribose, and it has got the base. Differentiate between the different types of nitrogenous bases. The four bases in DNA are guanine, cytosine, adenine, and thymine. RNA has uracil instead of thymine. Guanine and adenine are double-ringed. They are purines, while cytosine, thymine, and uracil are single-ringed. They are pyrimidines. Guanine and cytosine pair via 3 hydrogen bonds. Adenine and thymine or uracil pair via 2 hydrogen bonds. Compare the structure of DNA and RNA. We shall look at their sugars, base, and strand. DNA has a sugar of deoxyribose. RNA has a sugar of ribose. Bases. DNA has thymine, guanine, cytosine, and adenine. RNA has uracil, guanine, cytosine, and adenine. The strands. DNA has a double-stranded or is double-stranded. RNA is usually single-stranded. List 3 types of RNA. We have messenger RNA or mRNA, an RNA copy of DNA gene, that is messenger RNA. Secondly, we have the transfer RNA, tRNA. This one transfers amino acids to the ribosome. And we have the ribosomal RNA or rRNA, the catalytic component of ribosome. Outline with the aid of a diagram how nucleotides are linked together to form DNA strands. We have the structures on our right showing how the nucleotides are linked together to form the DNA strand, forming the DNA ladder and forming a double helix. DNA nucleotides are linked by covalent phosphodiester bonds, that is between the 5-phosphate and the 3-hydroxyl of the 2 nucleotides to form a long polynucleotide strand. The 2 strands join together via complementary base pairing. Guanine pairs with cytosine via a 3-hydrogen bonds. Adenine pairs with thymine via a 2-hydrogen bonds. The 2 strands run antiparallel to each other in order for the bases to face each other and form hydrogen bonds. The double-stranded DNA then twists into a double helix. Each twist occurs every approximately 10 nucleotides. List the contributions of Linus Pauling to the elucidation of DNA structure. Pauling identified molecular distances and bond angles for basic molecular structures. List the contributions of Phobos Levin to the elucidation of DNA structure. Phobos identified structure of a nucleotide, that is, the sugar, the phosphate, and the base. List the contributions of Erwin Chargaff to the elucidation of DNA structure. Erwin determined that there is an equal number of purines and pyrimidines. List the contributions of Rosalind Franklin to the elucidation of DNA structure. Rosalind identified the organization of DNA into a helical structure. Describe how Watson and Crick proposed the structure of DNA via model making. Watson and Crick used the above knowledge from the other scientists and constructed the DNA model through trial and error. They demonstrated that DNA strands were antiparallel and formed a double helix. They also showed the complementary base pairing. Hello, my friends. Thank you very much for participating in Biology King. I encourage you to subscribe.